In this video, we will study about the process of glycolysis. The word of glycolysis made up of two words which are glyco and lysis. The word glyco means glucose and the word lysis means breakdown. The glycolysis is a metabolic pathway that involves systematic breakdown of glucose to produce energy. The glycolysis takes place in cytosol of the cell. First, we have the overview about the process, and then we will study about the reaction involved. In glycolysis, one molecule of glucose, which is 6 carbon compound, is broken down into 2 molecules of pyruvate, which is 3 carbon compound. In this conversion, energy is released in the form of ATP and NADH. The pyruvate form is further metabolized in energy pathway that produce more energy. The process of glycolysis takes place into two phases. In the first phase, the glucose has converted into 2 molecules of glyceraldehyde 3-phosphate. This conversion involves in the addition of phosphate. The first phase actually uses energy instead of producing it. In the second phase, the glyceraldehyde 3-phosphate is converted into pyruvate. In this phase, the process will produce energy. Next is glycolysis reaction. So, it all begins with a molecule of glucose that enter the cytosol. The glucose is phosphorylated by enzyme hexokinase to produce glucose 6-phosphate. The word hexo means 6 and kinase is the enzyme that catalyzes phosphorylation reactions. The enzyme hexokinase transfers a molecule of phosphate from ATP to glucose, and glucose produces glucose 6-phosphate. In this step, ADP is produced, showing that a molecule of ATP is used. Glucose 6-phosphate will then converted into fructose 6-phosphate, which is catalyzed by the enzyme isomerase. Fructose 6-phosphate will converted into fructose 1,6 biphosphate by the addition of phosphate group to the first carbon of the fructose ring. This reaction is catalyzed by the enzyme phosphofructokinase. In this step, another molecule of ATP is used. Fructose 1,6 biphosphate is split into two molecules, which are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These both are 3-carbon compound. This step is catalyzed by the enzyme aldolase. This is the first phase of glycolysis. As you can see, two molecules of ATP is used. The two compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, are actually isomerase. They can convert into each other by the enzyme isomerase. But since glyceraldehyde 3-phosphate is further utilized in the process of glycolysis, the equilibrium of isomerase is always towards glyceraldehyde 3-phosphate. Also, keep in mind that every molecule of glucose is split into two molecules. So, every reaction in the second phase happens twice and will produce twice of products. Glyceraldehyde 3-phosphate will produce 1,3-biphosphate glycerate. This reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. NAD is reduced to NADH, which is further utilized into ETC to produce more energy. This reaction utilizes a molecule of inorganic phosphate, 1,3-biphosphate glycerate into 3-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate kinase. In this reaction, ATP is produced. 3-phosphoglycerate will then converted into 2-phosphoglycerate, and this reaction is catalyzed by the enzyme phosphoglycerate mutation. 2-phosphoglycerate will then converted into phosphoenolpyruvate. This reaction is utilized by the help of enzyme enolase and magnesium ion. Phosphoenolpyruvate will then converted into pyruvate with the help of enzyme pyruvate kinase. In this reaction, another molecule of ATP is produced. This is the second phase of glycolysis. Next, we will take a look at the net reaction of glycolysis. First, glucose will converted into two molecules of pyruvate. Two molecules of ADP will converted into two molecules of ATP with the help of two molecules of inorganic phosphate. And lastly, two molecules of NAD+ will converted into two molecules of NADH.
In this video, we will study about gluconeogenesis. Let's split the term so that we can remember the process. Gluco means glucose, neo means new, and genesis means production. At normal events, carbohydrates are broken down by human body enzymes into glucose. This just basically process of breaking down. At special circumstances, substances like fat, amino acids, and pyruvate are converted into glucose. Thus, gluconeogenesis can be defined as synthesis of glucose from non-carbohydrate sources. Then we will take a look at sites of gluconeogenesis. 50 to 60% process of gluconeogenesis occur at liver. 40% process of gluconeogenesis occur at kidney. The process of gluconeogenesis only occur minorly at intestine. Next, we will talk about concept of gluconeogenesis. Every person consumes food daily that converted into glucose, and this maintain blood glucose level between 100 to 200 mg per deciliter throughout the day. The excess of glucose converted into glycogen, which is stored in the liver, and this is used later when the blood glucose level falls. Another fasting state, the blood glucose level start falling and excess the glycogen about 100 gram that converted into glucose, which maintain blood glucose level. This glycogen reserve soon get depleted and blood glucose level start falling. Some tissues in the human body, such as nervous tissue and red blood cell, can only utilize glucose and source energy. So, substances like fat, amino acid, and pyruvate can convert it into glucose through processed gluconeogenesis, which maintain the blood glucose level. Metabolic reaction involved in process of gluconeogenesis. The process of gluconeogenesis involves glycolysis, Krebs cycle, and some other reaction also. So it is very important to understand about process of glycolysis and Krebs cycle in details. Make sure to understand the process of glycolysis and Krebs cycle. It's 6 carbon molecule and if you have studied about the glycolysis, glucose is converted to two molecules of pyruvate, which is three carbon compound. This happens through 10 series of reactions in glycolysis. From these three reactions are irreversible. It is important to understand the three irreversible reactions, which are catalyzed by specific enzyme and only regulated when concentration of glucose is high, so that the excess glucose converted into pyruvate. This prevents excess concentration of blood glucose, but these three irreversible reactions can be reversed in specific enzymes, which are regulated only when there is deficiency of glucose in the blood. These reactions are upregulated during special circumstances, such as fasting. During fasting, the pathway can run completely in different reactions. When the pyruvate gets converted into glucose, other substances such as propionate first converted to intermediate of Krebs cycle, which then get converted to pyruvate, and substances such as lactate first converted to pyruvate directly and converted into glucose. We will talk about glycolysis. First, glucose is converted into glucose 6-phosphate by the enzyme glucokinase. Then, the glucose 6-phosphate isomerizes into fructose 6-phosphate. The fructose 6-phosphate is converted into fructose 1,6-biphosphate by the enzyme phosphofructokinase. And through series of reversible reaction, fructose 1,6-biphosphate is converted into phosphoenol pyruvate. And to the last of the reversible reaction, the phosphoenol pyruvate is converted into pyruvate by the enzyme phosphofructokinase. The first reaction, which is irreversible and we have talked about, is conversion of pyruvate back to phosphoenol pyruvate. For this pyruvate first travel to the mitochondria through specific carrier protein. In the mitochondria, pyruvate is converted into oxaloacetate by the enzyme pyruvate carboxylase. In the green are the gluconeogenic enzyme which expressed during fasting. In this reaction, carbon dioxide is used and ATP is also used. Oxaloacetate is transferred back to the cytoplasm, and this oxaloacetate will convert into phosphoenol pyruvate by enzyme phosphoenol pyruvate carboxykinase. In this reaction, GTP acts as electron donor and carbon dioxide is elaborated back. Phosphoenol pyruvate then is converted into fructose 1,6-biphosphate since the reaction are reversible. The next reaction, irreversible reaction, is fructose 1,6-biphosphate converted into fructose 6-phosphate by the enzyme fructose 1,6-biphosphatase. The last irreversible reaction is conversion of glucose 6-phosphate to glucose by enzyme glucose 6-phosphatase. Phosphatase are the enzymes which break the phosphate bond. Other substances like propionate, lactate, and glycerol also converted into glucose through specific pathway. Let's first talk about propionate. Propionate is first converted into succinyl-CoA, and you have watched the video about Krebs cycle, and you know that the succinyl-CoA intermediate of Krebs cycle to form oxaloacetate. The oxaloacetate converted into phosphoenolpyruvate. Next is substance like lactate. Lactate is converted directly to pyruvate by the enzyme lactate dehydrogenase. And lastly is glycerol, which is derived to fat tissue, is the first converted to glycerol 3-phosphate, which is then converted into dihydroxyacetyl phosphate. And dihydroxyacetyl phosphate can be converted into fructose 1,6 biphosphate to the reaction of glycolysis. So this is all about gluconeogenesis. I hope this video can help you. Thank you.